start recording, so don't say anything you'll regret later. And if I noticed there were a few other people who raised their hands, feel free to drop it in the chat. I'm happy to answer any lingering questions that may not already be answered in the syllabus. I'm great. Thanks for asking, Fawad. I am doing dandy. Yeah. How are you? It is cold. Yeah. Yeah. No, I like the cold and I like the winter. So for me, I love it. Exams will not be in person. They will be done over Blackboard. It is so Nafisa says, isn't this supposed to be a hybrid class? It is a hybrid class. This part is online. the recitation part will be in person, where you meet with the TA within the class, and they will go through problem sets with you. And it should definitely help with your learning of the material in the course. So that is the hybrid part. Unfortunately, I'm in a situation where I am, as a lecturer, restricted by when my kids are dismissed from school. And the situation is both for myself and my wife, who are both professors at Queen's College. So we're pretty much at home. And for what it's worth, and it's probably fair warning, it is recording now, so I can let you guys know. I have 4 kids. We have 4 kids. So they're very loud and all over the place, especially at this time of day. So I try my best to restrict the teaching that I do, where it's important that you hear me during a moment when I'm audible. So I'll typically do that in my office. I might record some asynchronous lectures in my office. You're welcome to attend those. If they fit in your schedule, I'll be happy to let anyone know when that does happen. But just so you're aware, because we're living in a post-pandemic or during pandemic scenario right now, I've got kids and they're everywhere. They're hard to get rid of. Not that I want to. They're delightful. Good afternoon. Good afternoon, Adrisha. Thank you, Marisol. So the lectures will be remote. Recitations will be in person. Yep, definitely. And I tried to send it out in an email earlier today. Perhaps maybe you received it, maybe you didn't. Can you tell them I said hi? I can definitely tell them you can say hi. They will most likely come down here at one point and introduce themselves, whether I want to or not. So I was in campus in Rumsfeld Hall, room 100. I forgot to check the announcement. Oh, I'm so sorry about that, Tanisha. I'm hoping that it's a bit clearer now, and you won't hopefully find yourself making unneeded trips to campus by any chance. Just because, I mean, honestly, the less out there and less interactions we have, personally, I think is the better. I wouldn't want any of you to get sick just trying to come to class. It's not worth it. Has a connect code been sent? Yes, it is. It's in the syllabus. Exams are not in person. Only the recitations are in person. I will talk about that now. Do you need a text? Oh my God, so many questions, so many answers, and it's only 5:01. Good news, everybody. We can move on to the lecture. I'm hoping that most of your questions and lingering concerns will be answered in the slides that I prepared, which are also on on Blackboard. I'm hoping to clarify all of these. So I'm going to stop at a few times during the lecture, and then when I stop, feel free to drop your questions. I'm going to say I'm stopping now to answer your questions. I'm not going to scroll up because who knows how many people are going to reply in the chat. But what I'm going to do is I'm going to take a break from the chat right now. I'm going to go into the lecture, which I take it you can see right now. Yeah. And let me just double check. You can see that, correct? You can see the lecture? You can see the Welcome to Biology 285, 585 Principles of Genetics Spring 2022? Yes, fantastic. So what I'm going to do is I'm going to go through a few of these slides. I'll take a break every now and then to return to the chat to see how you're all doing. And hopefully, if there are any lingering questions that are not answered, I'll be able to answer them. So welcome. It is a very warm welcome that I greet 2022 with this class, first class of the semester in genetics. There's a lot to cover and not a ton of time. So We're going to go over the syllabus for the most part, or I guess very salient, important parts of the syllabus, so that way there's no confusion. You may hear about jokes about professors talking about read the syllabus. Did you check the syllabus? Read the syllabus. All of your answers will be answered in the syllabus. You know what? That's because all of your answers will be answered in the syllabus. So definitely read through the syllabus. Take the time and moment. Don't. Wait, hold your questions. I won't be able to see them. The reality is I won't be able to see them until after I take a break. But please take a moment to read the syllabus. I'm only going to go over a small part of it. The rest of it is your responsibility to go over, and a lot of important information is there. So don't miss out on it. I am your instructor. My name is Sebastian Alvarado. You can call me Professor, Dr. Alvarado, or whatever. And obviously, if you're in this class, you know that our class is from, let me just I want to make sure that we're recording, just because the worst thing can happen now. We are, okay, good. Mondays and Wednesdays, 5 o'clock. Typically, our class will be an hour and 15 minutes. I shouldn't go over that. I have the Blackboard set up to go up to half hour afterwards if there's any lingering concerns that need to be addressed after class. Office hours are by appointment only. So if you need to make an appointment, you'll see in the syllabus, I have a little link, or you can contact me via email. You are welcome to contact me until I respond. Usually within 24 hours is what I try and get emails back out by. But if I don't, after 24 hours, if you don't hear from me, you are invited and welcome to contact me again. Often when I take a break away from email for maybe 10 to 15 minutes, I come back and I realize I received 20 to 30 emails in those 10 to 15 minutes. So I am not ignoring your emails. It is not at all personal. It's just very difficult to manage so many requests all the time. So definitely reach out if you need to. And if I don't get back to you within 24 hours, reach out again and repeat. Hopefully you won't have to do that more than once. The textbook is specifically Hartwell Genetics from Genes to Genomes, 7th edition. And this is a book that we have to get through Connect or McGraw-Hill. But before we get into that, I'd like to just ask the class a few questions and flip back and forth. Perhaps we can start off by just asking how many of you are seniors? I'm now on the chat. So how many of you are seniors? Me. How about this? Give me a thumbs up if you're a senior in the chat. Thumbs up emoticon, thumbs up emoticon, thumbs, no, not me. I don't understand me. There we go, Basma, you know. Rudaba is a senior. Okay, I see a lot of thumbs up. Okay, it's quite a few. And then if you are juniors, can I see, let's see, can I see a heart? Because Valentine's Day is around the corner. If you are a junior, look at all those hearts. There's a handful of you. Okay, oh, quite a few. Always good to know. This is more fun than definitely putting up your hands in a lecture hall. Okay, now let's see. Any of you sophomores? If you are a sophomore, you have to pick. Ooh, alien head. There we go. Let's see. Any sophomores? Just more hearts. Oh, there's a few. Not as many. Or maybe. Okay. All right. Cool. In biology, so many of you may or may not be, but if you're in biology, emote a cat. Let's see, how many biology majors do we have here? Whether you're a sophomore, or junior, or whatever. Antonio, Aria, Maria, Jasmine, Marisol. Oh, there's quite a few cats, biology majors. That's good to know. It makes sense. It's a genetics class. All right, any chem majors? I know they sneak in every now and then. Oh, there's got to be a chemistry emoticon. No? Is there? Let me see. I'm sorry, I have to find out. Perhaps not. Pick this thing. Evil monster thing. Okay, some chem people. There's Deanna. She's in chem. Nilsa. Okay, very cool. And then anything else that wasn't necessarily mentioned. So maybe psych, history, politics, and pick whatever emoticon you want for that. Maybe not alien, but if there is, sometimes you have biochem. Okay, clown. Okay, cool. There's a bit of a variety. That makes sense. Biochemistry, I get it. Becoming very cool. Emily's going to be a bio teacher. And of course, the graduate students, I haven't forgotten about you either, actually. So feel free to chime in the chat. I know there's maybe 6 to 8 of you. So definitely feel free to let us know if you're a graduate student, if you're taking it for 585. Okay, Emily is. Very cool, Alex. All right. So some of you are teachers. Some of you might be planning on getting a PhD. Honestly, I'm happy to just skip through these. I don't want to spend too much time, but it is a good idea to get a survey of the class in general. If you want, maybe just chime in whether you might be interested in a PhD, an MA, if you're pre-med, pre-dental, or PA school, or maybe nursing, or maybe teaching. All of these are different options. I'd love to get an idea. I know we get quite a few clinical interests at this for this specific course, so it always helps to kind of measure expectations accordingly. So a lot of PA school, a lot of pre-med, nursing, PA, DPT. I'm sorry, what does DPT mean? I'm less familiar with that one. Doctor of Physical Therapy. Ah, okay, very cool. Excellent. So there is obviously very clinical interests here. Pre-pharm, right, pharmacy school. I forgot about that. There are quite a few clinical interests. What I do my best to do is I have to start off by saying genetics is a course that was typically, it starts quite often in the realm of the model system dictating the type of science or the kind of discoveries that we made over history. So there's quite a bit of a historical context and a focus on things like yeast, fruit flies, and worms. And I know those aren't clinical. It's not a human, right? It's not a human brain or a human kidney or liver or anything like that. You might find more interesting, or a human interaction. But there is quite a bit of historical context with those model systems. And I'm hoping that we can talk about that and explain the relevance and the importance of these things in basic research and connecting them to clinical setting as well. So just to kind of measure expectations, I've often talked to students who take this course and they're like, oh, I'm so sick of talking about fruit flies and mold and other things like that. And I relate and I understand because I'm teaching it, and I'm also perhaps not always thrilled about things that don't have spines. I like studying vertebrate systems, or at least interesting invertebrates, and often it can be difficult, but I will do my best to make it as engaging and exciting as I possibly can. So a little bit about me. Like I mentioned, my name is Sebastian. I was born in California, but I lived pretty much most of my life in Canada. For my undergraduate, I went to Carleton University, where I was going to be a performer. I was a magician, juggler, and fire breather. I did a lot of busking. My parents were horrified that I might run off and join the circus, but I did find myself going to McGill University, where I was doing a lot of cancer research. The cancer research was interesting, but I found myself interested in other model systems a little bit more. And I kind of mentioned, and I touched on this, that fruit fly, bread mold, and yeast, and these things can be kind of not as captivating as other model systems. And I found myself drawn to other model systems, specifically systems that can change under various environments without changing their genes at all. So I actually am in the field of epigenetics, or basically how your genes, or genes in general, speak to the environment, and how the environment speaks back, and that conversation and how it shapes different traits. So here I looked at how different animals change their size, and I also looked at ground squirrels to see how they hibernate and the type of molecular changes that accompany hibernation. I then did my postdoc at Stanford University, where I studied this fishy here. It's an African cichlid called the Statotilapia burtoni. And I studied them for a lot of reasons. One is their behavior and the neuroscience that comes with this system, but also they're really cool patterns and colors. They change color. They change from yellow to blue. They also change these patterns that you can see here, this pelvic fin stripe, this humeral blotch. They can actually disappear depending on where they are in social status. And then there are these little anal fin spots and all kinds of dorsal changes that allow them to be cryptic, but also to send social information to other fish. So I found myself very interested in this system. when I was a postdoc, and then I started my lab here at Queen's College. And since then, I've gone crazy. In addition to this system, which we actually have on the third floor of the science building, we decided to adopt the sunfish. This is a fish that's actually very common in North America, all over North America. Fall armyworms, these are cannibalistic. They'll eat each other if they are given the right cues. And we have two other vertebrate models vertebrate mammals that we're working with tissues and collaborators in Canada and in Scotland. And we're working with Tyler Stevenson, looking at Siberian hamsters, and with Rudy Boonstra in the Yukon. Well, he's in University of Toronto, but we're looking at how their colors change. So these two systems here, we're looking at how they change a summer for a winter coat, and they change white. And these guys we're studying for their cannibalistic tendencies. And these guys are actually very similar to our cichlids, except instead of these guys coming from Lake Tanganyika in Africa, in bordering Zambia, where there are lots of crocodiles, these guys are just in like upstate New York, a lot easier to get to, not as dangerous. My wife doesn't worry as much either. So it's much easier to do field work with these guys. Although we do field work with a South tilapia bird and I as well. We have plans to go up to the field in Zambia later this year. As I mentioned, I study epigenetic processes. We will touch on them in this course. Specifically, I look at how DNA, when it's stored inside the nucleus of a cell, if you unwind it and unravel it into this little fiber and remove all of the proteins that it's bundled up in, you can actually see that there's this modification called DNA methylation. And we study that process and how it regulates gene function under different kinds of environmental cues. We study this in our fish system here, which is a member of a very large family of animals that evolved very quickly in these three lakes, Lake Victoria, Lake Tanganyika, and Lake Malawi. And some 2,000 species evolved in about 10,000, or no, sorry, in about a million, about 3,000, 2 to 3,000 species evolved in about, I think it was 10 million years, which is kind of like an evolutionary eye blink. It was very, very quick. It's one of the largest adaptive radiations that's ever occurred among vertebrates. In addition to this, we look at their behavior. This is an example of our blue morph and our yellow morph, and we found that depending on whether they change blue or yellow, it also changes their behavior. Males that are yellow are often more aggressive and threatening. They do this threat behavior called a side display, which you can see right here, or a lateral display. And males that are actually blue spend more time going into the pot and holding their territory, which we think is reproductively motivated. Very quickly, we look at these behaviors, we watch them, we score them, and we turn them into really cool data that predict the patterns of behaviors that we think are occurring. For example, in our blue male, where we see more of these pot digging and entering, we see a sequence of behaviors all color-coded in green that indicate that this animal is probably reproductively motivated. It's interested in courtship. You'll notice that the same collection of behaviors isn't very connected to themselves and happens at a much lower frequency depending on the thickness of this arrow within yellowfish compared to bluefish. So basically our bluefish seem to be more reproductively motivated, but they also seem more conflict averse. And we use this model system to understand a lot of different things. We use it to understand how genes regulate behavior, how genes regulate color. And this has actually garnered some interest from, of all places, the United States military. We use this as a model of social adversity, and we use it to model aspects of social networks, individual behavior, and the molecular changes that accompany these things. In fact, we got money from the United States Army to specifically look at this and how it changes DNA methylation in blood. But that's not all. Like I mentioned, we do some field work. This is us in the field, just in Byram Reservoir in upstate New York. And I'll show you really quickly a video. I don't know if you can see it, but let's start it right now. These are some high school students and a post-bac student that's in my lab right now. And this is actually some, I don't know, 10 feet from the shoreline. You can see that this is a sunfish. They hold these territories, these little middens that you might see if you look far enough or you pay attention to them. And essentially there's a male here that sits and camps out. You can see he's coming in right here. And he tries to court females. So what he tries to do is wall her in. She'll drop her eggs and he'll release his sperm onto it. And I don't know if you saw that. There was a fish that just swam in there really quickly. The drama in the lives of these fish is actually rather interesting. That's a sneaker male, and he very quickly released his sperm onto those eggs when the other male wasn't looking, and he'll continue doing that, and other males will also continue doing that. They try and basically sneak in there, reproduce with the female and run out fast enough that the other male doesn't notice. Now, what's particularly interesting is this behavior initiates the dropping of eggs, which is a very protein-rich source of nutrients, and the female will leave them behind for the male to defend. It's his job to be a good dad, to take care of those eggs, and to keep all the other fish from snatching them up. And if you stay long enough, let's see if this, you'll notice that after she lays her eggs, she leaves, and all of a sudden, all of these other fish, same species, start to huddle around, and they eventually ambush this poor dad trying to take care of his young. And little by little, you'll see that they kind of amass, they precipitate, and then eventually they swarm and start eating all of the eggs in this nest. So this is what we're doing. When you see people at the side of a lake sitting in the water saying they're doing research, we're really interested in these kinds of behaviors, these social behaviors, the ones that determine community within a fish, and we try our best to translate that to other higher vertebrates such as humans as well. If any of this is kind of interesting, I strongly encourage you to maybe reach out to me independently to join our lab. But also there's a field course that I plan on teaching in July where you can actually get involved in this research. You can sign up. We're hoping to take only 12 people that can basically help us record these things out in the wild. We set up GoPros just on the side of the lake, sit in the sun for a bit, then we pick up those cameras and we take that data back home. We also catch these fish and we look at how it changes patterns in their brain, patterns of firing. So we look at how different dads at different parts in the territory differ in neural firing in known areas of the brain, and we look how it changes behavior. So enough of that. If that interests you, check out that field course and feel free to email me about it as well. So this is the textbook that you'll be using for the class. It's called it's called Genetics from Genes to Genomes. There's a 7th edition that you can get from McGraw-Hill. You are both enrolled in a lecture that has a recitation component to it. The recitation instructors are listed up here. There's Caitlin, who does the Monday sections, Joseph does the Tuesday sections, and James Carpino does the Wednesday sections. Everything, I believe, will be done online. Sorry. They will be done in person. However, if you are in a Wednesday recitation section, James Carpino is recently being onboarded into the Queen's College system, so he doesn't have access to campus just yet, but I do believe, again, if you're only on the Wednesday recitation, I believe he will be coming onto campus within the next week or two. So there is a chance that the Wednesday recitations will be done online through Blackboard, just like you're using Blackboard Collaborate right now. And in that case, it will only be for the short term. And soon enough, James will be on campus and you'll be able to talk to him in person for the recitations. In every recitation section, you will be expected to attend and take a quiz. That quiz usually covers information that was given in the lecture that week or the week before, I believe. And it counts towards 15% of your grade. There is assigned homework in the syllabus. You'll see at the very, very bottom of the syllabus, each chapter and each assignment and every question that corresponds to that specific assignment. If you complete each recitation or if you complete each assignment on time, you will be ready for the quiz. If you do not, you will not be ready for the quiz and you will do poorly on the quiz. And the quiz counts for, I believe we take the top 10 quizzes and it will count for a total of 15% of your grade. If you stay on top of this work, it will be great, it will be fine, and you will also be better prepared for the midterms and finals. I gotta stress, this is not an easy course. I do try my best to be as transparent as I possibly can to let you know what you need to know to do well in it. I truly, sincerely hope that all of you have it in you to get that A that you probably want for this course, but it is a very large commitment of time, and we'll talk about that a bit more later. But just for the recitation instructors, you have all of their information here. It's also in the syllabus. All right, let's see how everyone's doing. People are saying yummy. I don't know what that's for, but maybe it's because they're eating their young, perhaps. There are 2 sections for Wednesday. That is correct. There's probably a few sections in the morning followed by the afternoon, and you have to find out which one you're going to. All Wednesday recitations. Yes, when I said that, I specifically mean all the people on Wednesday recitations. Those are going to be done by James Carpino. you can maybe one day learn to clone yourself, Jared. I'm afraid I can't help you there. Are there openings to switch? Nope. Why is the recitation in person? Because it is, and it's hybrid. And so the class will be online throughout the semester. This lecture component, Mondays and Wednesdays at 5 o'clock, yes, it will be online throughout the semester. Are these slides going to be posted on Blackboard? Oh my God, I read your mind, Amaris. They are already on Blackboard. You just have to check. But yes, I will post all the slides on Blackboard the day of any given lecture. I won't post them the day before. I will post them on the day of. They're already prepped. I just think that everyone builds a false sense of security when they think everything is in one place. It's like, oh, I'll get around to it when I get to it, but I will give it to you the morning of. Wait, so there's homework listed? Yes, there is, Jonathan. It's on the syllabus. Can you talk a little bit more about the assignments? Yes, the assignments typically cover questions at the end of the chapter. There are a series of questions at the end of the chapter. I have picked a few of them to focus on, and that will be the assignment. You should be able to navigate them based off of what you learn within the chapter and the chapter readings, and that will help guide your effort. If you have any lingering questions about them, talk to your recitation section instructor. They will help you. They are there as a support system to ensure that you understand these assignments. So should they end up on an exam, you should be able to complete them. All right, hold on. I don't want to, okay, will the slides be posted? Yes, I already answered that. How much time for each exam component? The time that it takes to be in the course. So it's an hour and a half, I believe. I just have a question. Why was the lecture not in person? That is a question that I'm not going to answer because it will be remote. Tuesdays and Mondays will be in person. That is correct. I don't see anything on Blackboard. Well, you should be able to see them on Blackboard because, well, you should. You can email me if you're still not seeing anything on Blackboard. Will the slides be put up at the lecture? Yes. Oh dear, I can't answer these questions if everyone is chatting. So just let me get to the bottom before you press enter on your question. We have to study the chapters with the lectures you've studied. Yes. When will you start assigning assignments? They're already assigned. Check the syllabus. How much time for each exam? An hour and a half. Hey, professor, are mics disabled? Yes, they are. Just making sure. No, you don't have any technical difficulties. They are disabled for the entire class. The slides were not posted on Blackboard, but yes, they were. And I'm not going to go on Blackboard just to show you that they were. They were definitely posted there. Will the exams be off the slides, textbook, or both? Both. Are the homeworks due by Sunday? So let me get to that point. I'll get back to that, Swara. It's in the slides, don't worry. You stated there are 12 assignments in the syllabus, has assignment 11 twice. This might be a typo. I'll fix it, Jessica, don't worry. I don't either the slides on Blackboard. They really should be. Try looking at content and you'll see a link drop there for the slides. If not, I will make sure that it's there. How can we get Connect for free? I'm afraid you cannot get Connect for free. We'll go over that part soon. So the exams are in person. No, they're not. They are not in person. The exams will be on Blackboard. Quizzes and recitation sections, the quizzes given in recitation sections will be in person. But the final exams for this class and every exam will be in person or online. Online! Finals, exams, online. Last time I'm going to say it. The slides are not on Blackboard. Okay, everybody, how about we just say the slides are not on Blackboard for now, and I will check to make sure they're on Blackboard. But I do remember putting them up there. Okay. Only the syllabus. You know what? We'll put up the slides after this. I'll check again. Okay, content only. Yeah, no slides. Oh no, I like in-person exams. I'm afraid that is the situation, Namra. I'm sorry. No slides. Okay, good. Let's go because everyone likes to repeat themselves for some reason because they like hearing me repeat myself. Let's get there. I'm going to continue going through what we have. We went over recitations. There is homework. Okay. Every week on Sunday night, you will be expected to have completed the Connect homework. All right, that's going to be assigned probably after this lecture. They count towards 10% of your final grade. All right, in order to get this Connect homework and access to it, you have to use the link that is provided on the syllabus, and I also provided it here in these lecture slides. That will take you to the Connect portal. There is a one-time fee that you have to use to get the textbook. I believe it is $86. And you need to get that because it basically signs you up with their Connect system that allows you to do these homeworks and really keeping up with those homework problem sets, the Connect science problems, is essentially something that we've seen year after year really improves the grades of all of you because it really forces you within a structure that supports you and helps you learn the material on regular deadlines that are every Sunday. Essentially, it forces you to read the textbook, right, before that week's lecture. So you're prepared for that because you will be assessed for 10% of that on Connect homeworks. It's always due the Sunday, assigned at 11:59. And if you didn't finish it, you can send me an email if you have a very good reason and any kind of documentation regarding your health from a doctor or anything to that effect. It's all in the syllabus. And if anything comes up, you're more than welcome to ask for an extension if needed. But for the most part, I will expect any written documentation or any of those things within 24 hours of any missed deadline. And that goes for pretty much any deadline in this course. You have 24 hours to just transparently communicate with me both documentation and any need to push a deadline. The end of chapter problems. You will be quizzed on this for 15%, and this is what you will do in the recitation sections. You'll see at the very end of the syllabus, there are a collection of chapters that correspond to an assignment, and there are a series of questions that are assigned that you have to complete before your recitation section. You will then be given a quiz on that assignment, and hopefully if you did those recitation problems, you'll be able to do very well on that quiz. However, if you didn't, obviously you won't. Again, the Connect homework, the recitations, all of these things are systems in place to keep you up to date on your readings and what is expected in the course. Because if you can't do that, this is where it becomes very challenging. This is why I'm trying to emphasize this is not a light course. This is a very heavy course. You are expected to do quite a bit to stay on top of it. So please try your best to give the required time that's needed for this course. Recitation quizzes. The recitation sections are designed to give you practice and problem solving. You'll notice the questions are not just semantic recall. It's not just redefine or explain this definition. They're often problems that require you to think critically and to work through them. There's quite a bit of logic and math in them. So you would be expected to learn that logic and math, approach your recitation instructor, and work through those problem sets. They're not all very easy. Some of them are kind of challenging, and you're encouraged to the best of your ability to talk to each other and your recitation instructor to be able to present them. Typically, after you do the quiz in a recitation section, the instructor will take some of the more challenging questions from the assignment and do kind of a whiteboard or a chalkboard explanation of them, and they'll walk you through the problem. We will take scores from 10 quizzes to make up your final recitation grade. And this is really where you get to talk to either James, Joseph, or Caitlin about what you don't understand. So please use that time to your benefit. They're there for you. They're your support system outside of myself. All right, quick overview of the lecture and the syllabus. We're going to go over many topics just to kind of give you some context. We're probably going over a little over 100 years of molecular biology in about one semester. So try your best to understand the undertaking that is and how much work it is. We will always start with some sort of historical context of what we knew back then and how we got here today. We will talk about Mendel's principles of heredity, extensions to Mendel's laws and exceptions, talk about how we've been able to abstract the idea of Mendel's laws to actual locations in DNA and chromosomes. We'll talk about mapping, recombination frequencies, DNA structure, replication, recombination. This becomes a little bit more rote memorization. This is definitely logical problem solving, just to give you a general feel for the course. We'll then talk about the anatomy and function of the gene and dissection through mutation, so how we can understand gene function by mutating it. This also kind of gets a little bit half-half into, let's say, some memorization, but also a lot of logic and problem solving. And then we will have our first midterm on March 16th. And this will cover chapters 2 to 6. After that, we will then go into gene expression and specifically how we build genomes and sequence them, how we look at genomic variation, how we look at differences between prokaryotes and eukaryotic life, and gene regulation within bacterial life. Then we have our second midterm. You'll notice there's quite a few midterms and finals. There's 2 midterms and a final in this course, so there's quite a bit to study for. you'll have your spring recess, and then we'll have gene regulation prokaryotes, and then we will have the second exam. Just so you know, you have quite a bit of time to study for the second one. And then we will talk about gene regulation in eukaryotes. We'll talk about chromosomal rearrangements and diseases that arise from that, like cancer, and how to manipulate the genomes of eukaryotes, like things like transposases and CRISPR. And then we'll talk a little bit about cancer, which is a very nice case study in understanding all the ways in which things can go wrong when it comes to genetics. And then we have our final exam, which will occur between May 18th to 24th. All right, I heard a lot of pinging in the chat. Let's see. People are talking about how much the textbook costs. It's $90, so I can change that. It's $90. The thing is that you gotta go with the Connect Plus because it gives you a copy of the textbook, which you'll need anyways. But in addition to that, it enrolls you in the homework that counts for towards 10% of your mark. So I strongly recommend that you get it. How long are the homeworks? The homeworks are usually a collection of questions. You can actually check the syllabus right now and find out yourself. And if you sign up with Connect, you can check the end of the chapter programs questions, and you can write them all out and have them ready to do right now. So just check the syllabus. I got Connect for chemistry, so if I paid for that already, I just use the class code for biology, or I have to pay more. Pretty sure you're gonna have to pay a separate fee, MRAs, but I'm not entirely sure. I got the textbook for free. That's great. I'm glad you got the textbook for free, but you still have to sign up for Connect if you want that 10%. Is it mandatory to complete them, or it's recommended to help with the quiz? You could totally, so we're talking about the recitation assignments. You could totally not complete them, but I don't think you'll do very well in the quiz because usually the quiz is some sort of permutation of a question taken from those homework assignments. So you really should do the assignment. And also, I'm free to take any one of those questions, change a few of the variables, and put them on an exam as well. Now I got to worry about the access code. Don't worry, the access code is in the syllabus. It's in the syllabus. You need to buy it regardless. Same for me. Will we get to meet the graduate students? Yes, you'll meet the instructors. Caitlin is a graduate student. Joseph is a graduate student. I think James is an adjunct, but you can meet those people. Yes, you can. I have Connect for Chem too. That they see, yes, you do need to buy a separate one, a separate license. What is the term paper about? That is for graduate students only. So we'll get to that part. Just wait a second. But I have time to get into Connect and figure out any issues. Yes, you have time. I believe Connect gives you a 7-day free trial where you get to feel everything out. If you're worried that you may not be in the right course, definitely reach out to me. I'm happy to check to see if you got enrolled properly. But once that thing rolls out, I'll give you to the end of this week to figure it out. Yeah, there you go. It's a 2-week free trial, so you can do the first 2 assignments for the first 2 homeworks. But really, it is a structure in place to help you stay on top of the readings, and that's the really important part. All right, we're going to keep going. All right, 5:35. I hope I'm not talking too fast. So this is your link to connect to the Connect. You just click here, either in the slides, which I'm about to put on now. Maybe I put in 585, I don't know. But don't worry, right after this, I swear to all of you, I will put the slides online because apparently they didn't get online. If you have any issues with the Connect platform, you're welcome to CC me. But I would strongly recommend you start your claim or whatever issue you might have with McGraw-Hill Tech Support. If they don't get back to you or nobody gets back to you within 24 hours, because honestly, at this stage in this business, you should probably get back to somebody within 24 hours, feel free to contact me with your problem. I don't know how much I can help if you haven't started a support ticket on McGraw-Hill, but what I can do is I can push the sales rep that got us onboarded with this whole thing to fix your problem. And I've actually had a positive experience with them. They're pretty responsive. So if you have any issues like, question didn't load properly, document as much as you possibly can, take screenshots if you can, and just supply those to McGraw-Hill. And they often reach out to me to let me know that, this student totally got screwed on their one quiz and you should really give them a break. And I'll be like, okay, fine, let's give them a break, yeah? Okay, so there are 2 exams and a final. The first exam covers chapters 2 to 6. It says 1 to 6, but it's 2 to 6. We don't really do anything in chapter 1. And then the second one is chapter 7 to 12. And then the final, it is cumulative. I know some of you might be moaning, oh, it's cumulative, that's hard. It's cumulative and it is hard. It is challenging. Once again, this is not a light course. Both exam 1 and exam 2 will be challenging in their own right, and the final will cover all of that. So please do your best to stay on top of readings. Try your best to perform as well as you can on the first, second, and final exams, because that is really the best way to ensure success, just staying on top of the readings. There is no makeup. If you missed exam 1 or 2, it just gets tacked on to the final. So now you know. You can have a written excuse, but again, that written excuse has to have some sort of documentation. We're pretty hard line there. It's pretty clear in the syllabus. So if you start sending me emails and sending me pictures of, I don't know, I've gotten pictures of, of Spectrum vans outside of people's driveways, and I don't know how that is, like, I can't help with that. I have to follow what CUNY guidelines say are excuses. I try my best, but if anything comes up, do your best to let me know as soon as possible. Don't wait until the end of the semester after missing 2 midterms and saying, oh, here's a doctor's note for that time, and here's a second doctor's note for this time. I will not accept them, and I have not accepted them in the past. I will continue not accepting them. So please, if anything does come up that I need to know about that will affect your ability to perform on the finals or the midterms, let me know as soon as you know. You won't be able to do it, and we will go from there. Okay, a little overview of the exam. We are obviously living in a post-pandemic world, or current pandemic, I'm not too sure. I'm well aware that you have many resources available to you, and I recommend and strongly recommend that you use all of them. I have submitted my exams to check because why not have them there too? I will also give them to you on Blackboard. You can have them there too. If you have resources available to you, definitely use them to your advantage, because the reality is that if you ever need to learn anything about anything, the expectation is that you have access to some sort of, Wikipedia or whatever. So go ahead, open book. Now here's the reality. You still need to prepare yourself. And I don't want anybody to get a false sense of security saying, oh, whatever, I'll just show up to the exam. I'll answer all of the questions in multiple choice and all the problem sets without a problem, because I'm going to search every answer on Google. It's not going to work out for you. I've seen it not work out for many. Usually we have some 5 to 10 questions sampled from a random pool. I'll let you know right now, I spend quite a bit of time making that pool of questions. I typically use question bank questions that you'll find online, and then I modify them ever so slightly. So that way either the same answer is incorrect or some other answer out of the 4 is correct. I spend quite a bit of time designing those questions. I do it intentionally. You're welcome to do whatever you want and use whatever resource you can, but if it doesn't necessarily work in your favor. So the best way you could possibly do well in this course is often by preparing yourself. By preparing yourself, you'll be able to take this test or these tests, be able to do them in a timely manner that will allow you to both recall information you've studied for, and also answer them swiftly. Essentially, we've played with the equation of how much time you have with how much information you can process, and really metered that people who have studied and gone over the material typically will go through the exam at a better rate or a faster pace than those who have not prepared. And that's essentially the only way we can do these courses online. There is no, there is no compromise. There's no change that will be made to this format. As many times as you email me, as many times as you might ask, I'm just not going to change it. It's actually worked rather well, and specifically with the midterms that I've designed for the last 2-3 years. So you have some multiple choice questions, usually making up half of the exam, and then the rest of it will be short answers. These will be questions that will be paraphrased. A few variables will be changed, but essentially they will be the assignment questions that are assigned as your homework. And sometimes I do pick 5 out of 6, sometimes 6 out of 8. I will post previous year's midterms so you can take a look yourselves. And from that, hopefully you'll be able to prepare yourself best. Did I say there will be no makeup exams? There will be no makeup exams. Okay, so let me quickly go over grading for both 285 students and 585 students. The 585 component is for master's or graduate students at Queen's College. There's a written paper component there, but if you're enrolled in 285, you don't have to worry about that. If you're enrolled in 285, all you have to worry about is the first exam, the second exam, the final, your recitation quizzes, and your Connect homework. That will make up your entire grade. All right, that's the majority of you. A handful of you are signed up with 585. It is very similar in breakdown, except exam 1 will be worth 20%, exam 2 will be worth 20%, the final will be worth 25, your recitation and your Connect homework will be 15 and 10, and then we basically took 5 and 5 from exam 1 and 2 to make a research paper that you're responsible for. So basically, if you're enrolled in 585, in addition to everything else the class is responsible for that I've already gone over, you will also be responsible for writing a research paper. It's about 10 pages. 10-page research paper on a genetic disorder, and it is due on May 1st. You should reach out to me well in advance of March 28th asking about a specific topic or paper that you want to go over, or specific disease, genetic disease that you want to discuss. It counts towards 10% of your final grade, and every paper that is turned in late, 5 points will be deducted per day late. So please do not submit anything late. In addition to this, we run all of our written work through the Turnitin and a bunch of other things that they send to faculty for basically checking for plagiarism. So we will use that. So please don't plagiarize anything. Don't cheat. Don't be dishonest. A quick overview. You can basically pick any disease pathology as long as it has some sort of genetic underpinning. You'll have to explain the mode of inheritance. Is it dominant recessive? How penetrant is it? Is it X-linked? Is it autosomal? What it leads to, so what are the biological outcomes of a specific mutation, and be sure to include 5 to 10 resources or references that cover your interest in that specific disease. Please don't pick sickle cell anemia. I've seen enough sickle cell anemia papers, so I'll put an extra note saying you can't talk about sickle cell anemia. It's just because like it's so easy to get copied paper or whatever. It's just I strongly, this is why you have to reach out to me beforehand. Just reach out beforehand, before the 28th. By then, you should have picked one. I will be there to remind you if you haven't. And please just know sickle cell anemia. I've read too many papers about sickle cell anemia. Some important resources that will help you are PubMed and OMIM. Oh, what happened? Oh, I clicked on the link. Apologies. And then, of course, Your Genes, Your Health, and the Genome News Network. So these are all resources that you can use. How to get a good grade in genetics? The $1 billion question, okay? Spend time studying. In the syllabus, we talk about spending a good 1 or 2 hours per hour of lecture. So after this, you could spend 2 hours. In addition to that, you have a recitation section, 2 hours, and then on Wednesday, another 2 hours. So a good 6 hours a week of studying is often what we typically see is required to get a good grade in genetics, and we're talking AA+. But don't do it all at once. Don't think that studying, the day before, the weekend before an exam is going to help you. It will probably work against you. And if you do pull it off, you won't be very happy. And I wouldn't want you to be unhappy. Read the textbook, listen to the lecture, and discuss with peers. So you have to read the textbook independently. It is an expectation that I've said. It's in the syllabus, and I'm saying it to you now. Listen to the lecture like many of you are right now. It will be recorded. It'll always be recorded and available for you to listen to. And then discuss with peers. This is your opportunity in recitation. So use those recitation sections and any additional time you might need out of the recitation section to ask all the questions you need to ask in order to understand the material as thoroughly as you can. Be your own worst critic. So be able to reflect on what you did learn and what you didn't learn. So respond thoughtfully to feedback on recitation quizzes. You might not do super well on them. You might find that you had a lapse in understanding a certain idea or concept. Use that as a place to grow. Use that as a place to develop your understanding. Don't let them beat you down or anything like that. And just be honest with what you think you're with how well you think you're doing. I've talked to so many students who are like, I did everything you told me to and I didn't do well in the midterm. And I'm like, oh, okay, did you read the chapters? Well, no, I didn't read the chapters. I'm like, oh, okay, well, did you stay on top of the Connect homework? Oh, I haven't been showing up to recitation. Oh, okay. Have you been reading the book? Well, I read the first chapter. So I've seen this over and over again. I just want you all not to make those mistakes, all right? So please stay on top of everything. Again, the recitation, the Connect homework, the lecture, these are all there as structures to ensure that you're learning, right? They're support structures that not only are ensuring that you're learning, but they're also connecting you with people who know about these things to impart their knowledge so that way you can understand it. And inevitably, there will be a few hurdles where you're like, nobody seems to be understanding, recombination frequencies. That's okay. You're welcome if outside of the recitation section, and in the lecture, you've listened to the lecture 10 times about that one idea. You went to the recitation section and you asked all of your questions and it's still struggling. Send an email to me and just say, I tried the recitation, I tried this, still won't go through. I'll be happy to take the time. Of course, scheduling, if you schedule time and you email me asking for that time, and we can go over these things. But essentially, you got to be honest with yourself and your performance in this class. Another way, and another really helpful way of exploring this is basically understanding how well you understand it to explain to somebody else. Often the best way to learn is to teach. And if you can find yourself in either a study group, remotely, or whatever, I recommend remotely. I don't want any of you getting sick if any of you have Omicron or whatever form of COVID is out right now. I essentially think that some of the best ways that you can study is often by teaching and explaining the same idea to other people and trying to explain to different audiences. So these are things that will definitely help you do better in the course. Try to find ways to make it fun and relevant. I do my best to do this as well. Sometimes I succeed, sometimes I fail, but still try. So read chapters before class, answer the questions on Connect Plus. They always cover the content that will be gone over that week. Answer questions at the end of the chapter. It's already in the syllabus. You can start now if you'd like. And then, of course, the quizzes on recitations are based on these end of the chapter questions from the week before. So you'll have a lot of ways to catch up and stay on top of it. So you have a bad week, that's okay. There's usually some sort of system shared between the recitation sections, the Connect homework, and lecture that should hopefully keep you caught up. Okay, this is something I've done in the past for extra credit. I usually only give up to like 1 or 2 points on a given midterm or final. If you can come up with a fun meme that illustrates a specific idea in the course, I'm willing to accept them. But I must emphasize original. I've seen a lot of people send the same memes, and since I've been doing this for 2 years, everyone keeps trying to send the same ones to me, but I've seen them already. Or if you want to come up with a good TikTok or whatever, you're welcome to. None of this is mandatory, so I'm not asking any of you to do it. I'm just saying, if you are really keen on the material or you're having a lot of fun with the material and you thought of something that you thought was funny, you're more than welcome to share it and to see if you can get extra credit for it. These are examples of things that came up that came up in certain students in different classes. Single-stranded binding proteins holding up the DNA helix, which you can see here in this movie Birdcage, I think it was, or Birdhouse, I think, or jokes about mitosis and meiosis too. I really just like that some students think of these things, and if they come up with really fun ideas, I will be happy to give them the credit that they want on the midterm, and I often share it with the class or through social media afterwards. So if I promised you, I've seen all of them already. So if you try reusing an old one, or if it's just not particularly good, I apologize in advance, but I'm probably going to say so, and I won't give you the credit. But I really want people to kind of put their best ideas for it. But again, not mandatory at all. This is all just for extra credit. This is just one way to make it a little bit more fun. Okay, very quick overview. Let me just switch back, see if anybody has any questions. How are we all doing? Let's see. Please give this for free. Oh, memes. Hold on. Will I be posting review questions? I won't, but what I'll do is I'll post previous year's exams. How about that? Yeah, that will be plenty to work with. You can pick whatever citation format you prefer, Alexander. I just want to make sure that syllabus. Everyone's saying the answer's in the syllabus. Good for you. Let me just see if I'm missing any questions. Please give us for free. I'm sorry. Will you share them with the class afterwards? Absolutely. I adore nothing more than sharing stuff like this. We have to make the meme. Yes, you have to make the meme. You can do it as a single frame. You can do it as a TikTok. You can do it as whatever you want. Just send it to me and I will take a look, and I'll let you know probably relatively quick whether I'll give you the extra credit or not. And I'll do it for a maximum of 1 point on a given midterm or final. So you can submit up to 3 of them. Yes, definitely make it original. If there are calculation-based questions that ask us to show calculations, would it be possible to submit our calculations from our pen and paper instead of doing Blackboard on Blackboard? Yes, you are welcome to do that. That is a great question, Jan. There are a few ways in which I would handle that, and a lot of it is, I would say for starters, I do my best to make the finishing of the questions on Blackboard as concise and to the point as possible. So if I don't imagine there being a very elaborate calculation that you would need to do, but if you feel that you're writing way more and you're deriving equations that I didn't ask you to derive, but you think that that's what you need to show to establish that you're being assessed properly on the content, you're welcome to send me an email of a picture of that calculation the moment you have it. But submitting it anytime after, like if you submit it 5 minutes after you submitted the exam and the exam's over, I will not accept it. So just so you know. Do I curve? Oh, that's a question I've never heard before. Sorry, Mahmoud. I don't curve if I don't have to. A curve is an artificial inflation of grades. It also brings down grades too. And I typically never do. So why would I even think of curving if I have not seen performance in this class yet? Is there a recitation on days of exams? There is no recitation on the week of an exam. There are no recitation quizzes. Sorry, to be correct, let me clarify. There are no recitation quizzes on the week of a given exam, and you'll see that in the syllabus. Would you provide the answer key to the midterms you post from previous years? Yes, I will. You stated that the lectures will always be recorded and posted, correct? If that is true, would it be okay for us to miss the live class and watch? Absolutely, at a convenient time. Better yet, I won't even suggest at a more convenient time. I'll say speed it up so you don't have to spend so much time listening to me. As much as I like to talk, I'm sure it's to your benefit as it is also to me if I were in your seat. So definitely don't feel required to attend class. How many points do you give for original memes? 1 point per exam. Someone asks a question at 5:38. Could you please answer that? Did I answer it? I don't know. 5:38. Do you curve? I already answered that. 1 point for each submission. Thank you for clarifying that, Alex. I meant, do we have to go to the class on the days of exams? If it's a recitation, yes, you have to appear in person. If there's a quiz, there are no quizzes, but I bet you if you go to that recitation and you're doing some last minute studying, the recitation instructor will be able to help you, but it's entirely up to you. So are you saying the work that is needed for a question on the exam must be uploaded? Mahmoud, I'm personally preferring that you answer everything in Blackboard, but if you feel that you've written enough on by hand that you really think that I won't be able to mark it, otherwise, I encourage you to send an email within the allotted time of the exam. I'll consider it, but I really don't think there will be a need for it. Should we take extensive notes during class? Yes, Nafisa, you should definitely take extensive notes during class. There's a lot of material to cover. Thankfully, we really hone in and we focus on a lot of these ideas, and we think about it really thoughtfully. So there's an opportunity here to go over problems really in depth. I go over a few of them myself, the ones that'll be taken out of the homework. But yes, totally take notes. Should we take, okay. Emily, the exams are online. Yes, they are, Emily. Thank you, I wasn't sure, that's okay. My recitation would be after the test. That is true. I'm sorry for that. If you really feel a need to and you want to attend another recitation, send me an email. I'll be happy to accommodate. You scrolled back to 5:38. That's dedication. Thank you, Delroy. Thank you. Are we going to have to remember a lot of dates? Well, if you look at the syllabus, you won't have to remember very much at all, except for the material you need to do well in this exam. Do you recommend any tutoring for this course? Gabriella, that's a great question. I am not entirely sure. Everyone learns at a different pace. Everyone has a different expectation of what they think is a grade good for them. Some people just want to do well enough to get their degree. Other people want to do well enough to get into medical school and they realize it's competitive. That is not my place to decide for you. However, if you find yourself a week or two into this course struggling and you're doing everything, keeping up, you're finding time to read, you're finding time to do the assignments, and you still find yourself struggling, you're welcome to make an appointment with me. Send me an email saying, I'm really struggling. I'm trying my best to keep up with it, but I just can't. And I'm happy to talk about it, and I can make it a more tailored personal conversation with you so that way we can both figure out what you can do to do better in this course. I really, despite what some people think, I don't want anyone to do badly in this course. I don't take any pleasure out of that. I actually take quite a bit of pride and happiness out of people getting exactly what they think they deserve. If they work really hard for a grade or they were able to bump it up 5 to 10% than what they thought they would, that actually makes me really happy. So I do want to be there to support you for that. It's just there's so many of you. I really want to direct as many of you as you can to the recitation instructors, which are actually there for you in smaller classrooms to help you. So if they can't help you or you're struggling, feel free to reach out, send an email, schedule an appointment. We can do something on Zoom. You are not required to attend the lectures, but of course, catch up later. Yes, you can. How about attendance? I don't care. Attendance really only matters for the recitation sections, and that's mainly because there's a quiz, and you can't take a quiz if you're not present. Is there a specific circumstance where there may be a curve? Nope, I'm skipping because I'm not going to answer the question, but a curve. For nuanced questions about class content, would you prefer us to email you or email? Wait for your recitation instructor to go over the content. If they can't answer the question, definitely feel free to send an email saying, hey, we just went over that recitation section. I still have a question. I have a lingering, something. I can't go to sleep because I'm still wondering if this means that and that means this. Then you're more than welcome to email me. But definitely consider you're in a line of defense that first the recitation instructors are there for you and then me. But I am happy to step in when and where needed. Don't worry about paragraphs. There might be a lot of information in the textbook. What would you recommend us to focus on? The things that you mentioned in lectures. Use lecture figures and points, and use lecture legends as your anchor in the content. They will sink you into what is most important. If that anchor isn't enough, definitely read the chapter to get more context. And frankly, you navigating the material and the lecture will help you do well on those Connect homework assignments. So between those things, you might find the strategy that works best for you, that you can ultimately assess after that first midterm. And if you realize, hey, I got the grade that I thought exactly I would get on the midterm, then that strategy will work for you. If you did not, then consider reassessing and sending me an email. Thanks. Is partial credit rewarded? Yes, I give partial credit. I definitely do. If I see that you're writing down, like if you got one number off or you took down the wrong column, but you wrote down logical ideas that make sense, I will definitely consider partial credit. I'll add one more to that. People who write just the answer down and don't show their work, I get a little suspicious, and I typically expect you to show your work. And I will say that in my midterms. I will say, show how you would get from here to there. So anybody who what's the mapping frequency or mapping units between these two alleles, and they're like 7. And there's nothing else shown there, I give 0 or 0.5 if that is the correct answer. If it's the wrong answer, it's 0. But please do show your work. I do give partial credit. I've given 4.5 out of 5 for getting the wrong answer, but showing enough of the work to make it make sense, to show that you understood what was being communicated in class. Class time is almost over. You're right. We have 15 minutes left. Will you cover everything in the chapter? I'll do my best to cover most of what's in the chapter. You can use the figures as a guide. Just so you know, this was just the remaining, the remaining slides were just overview of what's going to cover in the lecture. Nothing that you have to worry about. So I'm happy to answer more of these questions if it's going to help you more. Prepare yourself for this course. Will you cover everything in the chapter? If not, would we be tested on anything you don't go over? Anything that is in the assignments, in the homework assignments, you can be tested on. So yeah, thank you. Yay, yay. So the recitation is considered the lab of this class. Yeah, it is kind of like the lab section, except it's more like problem solving logic. I mean, we only have 50 minutes left of class, so I would guess that we wouldn't go over chapter 1. Yeah, don't worry. Don't worry. What should we do before our next class? Hang tight. Read the chapters. Go through the syllabus. Read it on your own. There's a lot of information there. Some things I didn't get a chance to cover. Special accommodations. Many people might need special accommodations. They might require extra time. I accommodate almost all these requests, but you need to go through the special accommodations services office. It's in the syllabus. The email is there. Please ask them and do it as sooner than later. The worst thing that I've seen happen over and over and over again, which is why I emphasize this so much, is somebody maybe requires a little extra time or they need other aids. And what happens is that they only find out after they did poorly on the first midterm and they didn't realize that that was an option. And it's very tragic and very sad. And I am really unable to help and I am out of options. So it's really important that if you need any of those special accommodations, you seek them out now and send me all the information I need to know. Typically, a week before midterm also helps, but you can also send it to me now if you'd like. The homework is already assigned, Zwaria. I hope I'm saying that right. Zwaria, is that right? Okay, good. Zwaria, yes, all of the homework is assigned. It's at the very end of the syllabus. You can go check it now and you can start doing it. Should we prepare chapter 1 and 2 for next class? Yeah, start reading the chapter. Start reading the textbook. Sign up with Connect. All of these things are things that you can do. What is something that has worked well with previous students that you would advise we do that what you've advised us in the PowerPoint? So what else am I missing in helping you help yourselves? At the end of the day, you have to just I've tried to give all of the information that you need to do well in this course. So everything I've set up to this point, I'm really pushing you towards like that A+, but perhaps maybe the A+ isn't what you need or what you want right now. I mean, as nice as it would be. Perhaps you're just kind of like, I'd be happy with a B in this class. Realize that how you meter your expectations will affect how much time you commit to it. So if you're putting the full 6 hours a week, you're doing every single question, you're finding every single resource and you're using it and you're staying on top of it, I hope that you find yourself at the upper end of the GPA scale. As time starts taking away from those things, I know many of you probably are working part-time, full-time even, and there's all kinds of other variables. For example, maybe you have kids. I know I have kids. I can totally relate. All these things make it more challenging, and I understand that these are the challenges that you face and that others face as well. I do my best to help where I can. There are some things I clearly can't help with, other things that I can. And when it comes to just developing a strategy, frankly, sometimes it's better that you approach me after a week or two of trying what does work or what I did communicate with you works, and then just let me know, like, listen, that's not working. And then I can just go one-on-one with each of you that are really feel like you're falling behind in that area. There is no homework assignment on Connect, but you know what? There will be soon. And when there is, I'll send out an announcement. Is the only homework we have and are graded is the Connect? Yes. Yes, really challenging. I hope we can all do well. I hope you can all do well in the course too. I'd be very happy to see that. Okay, cool. So let me just quickly go back. And honestly, what is genetics? Just a quick overview. I know we've got 10 minutes. It's a study how genes control characteristics, traits more generally, and they are hereditary. So you inherit them across generations. Genetics as a field is how we study genes and how they control specific traits, how they behave, how they're regulated, how that information is stored, and how it's transmitted across similar or related organisms. So when you're kind of looking at evolution, that's a very important area of genetics. We will talk quite extensively about the structure, function, variation, distribution of genes and how they change. They became so important in the mid-1950s or in the mid-1900s that they became really the only molecular substrate that we could study to understand biology. Biology was a very descriptive field. It was about, drawing flowers and pictures of cells. Not that there's anything unimportant about that. It's really, really important, but it was microscopy and our understanding of biology was really just that for several hundreds of years, up until, when we started looking into genetics and we started finding the molecular variation or the molecular changes that can explain biology. So we typically describe a gene as a locatable region. It is something in measurable space within a genome, and it corresponds to something involved in inheritance. It has its associated regulatory regions, parts of it that get transcribed, which we'll talk about it, don't worry, and other functional roles, other functional sequence regions. We're going to talk about DNA, RNA, protein, and how these things integrate in biological systems, how these things rely on one another. Some basics, we're going to talk about structure and how that relates to function. So specifically, how structure of DNA allows it to base pair and form the double helix. We'll have whole lectures dedicated to that. We'll talk about where DNA is stored, how it's stored in chromosomes that exist in the nucleus of any given cell. You can imagine that, some 3 billion base pairs of complexity are individually stored within one given nucleus. And if there's 37.2 trillion cells in your body right now, that's a lot of DNA, a lot of information. And that information is used in a very context-dependent way. We'll talk about RNA. We'll talk about RNA as the product of DNA templates that transcribe to make RNA. Don't worry, I may sound like I'm rushing over it right now. The only reason I'm rushing is because we'll talk about it in depth. We talk about how that RNA can encode protein and amino acids that make up a lot of the, doers of biology. So the specific molecular machinery is known as proteins that can have specific jobs in a given cell or tissue. We'll talk about proteins and how they're made up of, how their structure determines their function. And here's a few examples of how their structure can vary through amino acids and different types of structures, and how if you abstract long sequences of these amino acids, you can get all kinds of alpha helices and beta pleated sheets and things like that, that represent the diversity of different proteins and their functions. RNAs are also very diverse. If you like that part, I recommend you take the advanced molecular biology course that I also teach in the fall, where we talk about, RNA modifications and RNA splice variation. And then we're also going to talk a little bit about evolution, about how RNA is some of the earliest information processing molecules, how RNA was also some of the first proteins, what we call it, well, not proteins, but like the first ribozymes were the nucleic acids that folded and had their own known functions. And then how DNA took over that role in later evolutionary terms. And how proteins also took over. So how RNA kind of started the fire, but then DNA and protein took it over. We'll talk a lot about homology. So here's an example of a gene that's conserved across yeast, a weed, a worm, a fruit fly, a mouse, and a human. This is a cytochrome C protein that's involved with detoxifying toxins from your body and your blood. You can see how all of these highlighted areas correspond to conserved homology, something that we call when it has stayed the same over millions and millions of years. We use this to study and understand genes and how they function. We talk about how genes increase their number across a genome, how they duplicate, they diverge, and they become new genes with similar structure but completely different functions. A really cool example of this, one that might be very familiar with each of you, is your hemoglobin. There are alpha, beta hemoglobins that all have different functions. Some of them are expressed when you are a baby, others are expressed when you're in utero, and some are expressed even in an adult form. Another really cool story about this is venom. Perhaps you've heard of venom, not Tom Hardy venom or Topher Grace venom, but venom, the venom that comes out of a scorpion's tail or a cobra's bite. Venom is actually the product of several normal genes that were duplicated and then mutated over time and concentrated within venom glands. So that way, when they bit something or someone, they would transmit a cocktail of proteins that have very broken functions, and it's through those broken functions that they become venomous, and then they can disrupt your cardiac and your cardiac and your nervous system. In fact, if you really like that stuff, I recommend you check out Monday Holford's lab at Hunter, because she actually does research with cone snail venoms and all kinds of other venomous animals. Anyways, small aside based off of how genes arise. How do we typically study these things? Oh, sorry, I'll take that slide out. We'll talk about sequencing genomes, how we've started with these very fundamental model systems: bacteria, yeast, worms, plants, fruits, fish, zebrafish, and mice. But this is kind of like the old guard of molecular biology. Since then, it's gotten way more complex. We study cichlid fish, fall armyworms, Siberian hamsters, and so on. And I'll definitely try my best to make things interesting to that, as well as relating it to human studies. We'll talk a lot about homology near the end of the course. Nothing particularly important. And then here we have one last example, kind of commenting on the translational importance of understanding genetics. Something like microcephaly can actually be resolved down to mutations in specific genes. It's a rare brain malformation, and we can discover what causes it by looking at the sequence of specific genes, such as WDR62, and where a mutation can get introduced. In fact, the deletion of these 4 nucleotide letters, so the deletion of these 4 letters in the book of life that makes up your genome, can create a catastrophic change in the proteins that are very important during development and can actually lead to this specific phenotype. And there are a range of mutations that actually find themselves, each labeled here, 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 and here, throughout this very important gene, that if you delete it or if you affect it in a way that it loses its function, it can lead to this microcephaly outcome. And it's 6:13. We have 2 minutes to spare. What do you think about that? And if you have any questions, I've got 2 minutes to answer them. Otherwise, I'll wait for questions. And in the meantime, I will try my best to upload these lecture slides, because apparently they didn't get up there. For post-chapter questions for the recitation, is there a way we can check if our answer is correct? Yes, the recitation sections will be able to solve all of the problems for you. And I believe they have solutions manuals that they can use. I don't want to study for my homework, for my quiz, if my answers are wrong. Totally. That's why it's important to talk to your recitation instructors. A lot of times, and I remember running recitation sections when I first got here, a lot of the time people just feel like they have to show up. You do to write the quiz, but you realize the quiz usually takes 5 to 10 minutes. It's a 5 to 10 minute quiz in a 1 hour slot. The remaining 45 minutes, they're yours, and they're yours to ask the recitation instructor your most lingering questions about genetics. They're there to help you. So please use their time to your benefit. It's there for a reason. Sorry, everyone's talking at the same time. Someone asked before for the post. Okay. Thank you. You're welcome. I sent an email earlier. Just wanted to let you know that you could take a look and get back to me. I will take a look, Emily. I'm sorry I missed that. I'll try and take a look. In fact, I would recommend, Emily, if you want to just re-forward it to me, that might be the easiest way, because a lot of you probably emailed me just now. Thank you for sharing about your life. Oh, my pleasure. And what you studied. Have a good evening. You too. Have a good evening. You all have good evenings. Oh, you're all so friendly and nice. When will the Connect homework? Probably within the next 24 hours. The next 24 hours, the Connect homework will be uploaded. Everyone's saying good night, have a nice day, good night, wonderful evening, and I'll take care. Yeah, we're done. So any lingering questions, just email them to me. Everyone have a great evening. All right, take care, stay safe. Stop recording, and then I'm going to step out.